Good morning, everyone. My name is Athanasia. I am responsible for pharmaceutical cannabinoids with dsm-firmenich. There we go. And I am delighted to be with you this morning to do the first worldwide presentation of our clinical trial, a trial which compares our novel CBD formulation for solid formats called CBtru to Epidiolex. So it's a phase 1 pharmacokinetic clinical trial with a clear focus on bioavailability. So in the next 15 minutes, I would like to take you through 3 points. First of all, what's the challenge we're trying to address? Secondly, the R&D journey we undertook with CBtru. And thirdly, of course, the results of the clinical trial. But before I do that, I would like to say a couple of words about dsm-firmenich, the company behind this innovation. It's not a name that many people know because we are a B2B company. We're the world's leader in ingredients for health, nutrition, and beauty purposes. And our mission is to bring progress in life by combining the desirable, the essential, and the sustainable. With about 150 years of heritage and over 30,000 employees across the world, we are fueled and backed by science. And that's just not words. When I say backed by science, I mean it. There are about 2,000 scientists and engineers working across our 15 R&D labs and 78 application labs. We invest about 700 million euros in R&D every year. And as a result, we have the largest ingredient portfolio in our industry and about 16,000 patents. So part of this scientific backbone of dsm-firmenich is also the Pharma Solutions team, on behalf of which I am here today. So in the Pharma Solutions team, we offer critical APIs, specialty excipients, but also regulatory and technical expertise to help pharmaceutical companies, to help you deliver your drug therapies and meet the needs of the most vulnerable population groups. And obviously, this is in line with the work that we do in the cannabinoid space. So let me go into that first of 3 points I mentioned. What is the challenge we're trying to address? So as you all know, since the discovery of the endocannabinoid system in the 1980s, research into cannabinoids has increased. We know that the endocannabinoid system interacts with many important aspects of our physiology and our mood, including appetite, sleep-wake cycles, immune responses, inflammation, and so on. And that cannabinoids, and in particular CBD being the most common among them, interact with the endocannabinoid system by binding to certain receptors, mainly in the brain or the immune system. And particularly in this last decade and the medical cannabis market's opening, we have seen that research, both publications and clinical trials into CBD, has grown exponentially. As of today, there are more than 220 clinical trials with CBD that are either completed or ongoing. And there are more than 24 indications for which CBD has shown therapeutic potential, including central nervous system disorders, pain, insomnia, anxiety, and many others. But important as it is as an innovation, challenges still remain when it comes to innovating with CBD. One very important challenge we all face, and I heard it being mentioned multiple times during these 2 days of the conference, is the low and very variable oral bioavailability of CBD. We know that orally absorbed CBD is about 6% in the human body, and as a result of incomplete absorption in the gut and important significant pre-systemic elimination in the liver. It is estimated that about 75% of orally absorbed CBD gets lost through hepatic metabolism. So we do need new formats that are able to deliver higher bioavailability, but also more consistent, because one thing we know is that there is significant variation between patients who may be taking the same dose, but are actually benefiting very differently. So this is all a result of the highly lipophilic nature of CBD, and as a result, what manufacturers have decided to do is deliver it in a carrier oil to improve the bioavailability. and deliver it also with a high-calorie meal, right? We know that it increases bioavailability, it increases absorption. However, what we hear from patients is that taking that oil is not always easy, it's not always convenient, it's hard to measure sometimes, the taste is not pleasant, and also we are not always getting the desired effect. So we decided this is a problem we want to help address. There is potential for CBD formulations that deliver higher and more consistent CBD bioavailability, an improved taste experience for the patient, and a consistent clinical response. And we do think that the aspect of taste, convenience, patient experience is important, because if a patient has difficulty taking their treatment, they're not going to stick to their treatment program, and no treatment is effective when you're not taking it. So let's move to point number 2, the R&D journey we undertook with CBtru. So this is a long journey. It's been more than 2 years that our application and formulation scientists have been working on this innovation. And they started by screening technologies across 4 different areas, which you see at the top of the slide. So on the left-hand side, you can see technology areas that we have expertise in-house, such as the spray-dried powder solutions or the amorphous solid dispersions. But we also did a lot of external collaborations in the space, as the amorphous dispersions you see here on the right-hand side, but also working on liposomal delivery, which we know is a common way to improve the bioavailability of fat-soluble actives. So our team took more than 200 prototypes through this technology benchmarking. And out of those, 120 moved to powder characterization and in vitro bioaccessibility screening. And out of those 120, the 6 best performing prototypes moved into an animal study, leading up to CBtru, our asset that went into a human clinical trial. So I'll give you a snapshot of each one of these steps, and I'm happy to take questions later. You see here a snapshot of our results from the in vitro bioaccessibility study. I'm not going to show you all 120. I don't think that 20 minutes we have is enough for that. So I chose the 6 prototypes that eventually went into the animal study. So if you look from left to right, you see the amorphous solid dispersion. Then you see in yellow the nanomulsified spray-dried solution. That one is CBtru, so that's the one we're going to talk about for the human study. And then amorphous hot melt extrusions, you see 2 because we use different excipients. Liposomal deliveries, solid lipid nanoparticles, and on the right-hand side, in blue, you see the comparator, which is CBD dissolved in DMSO. One point I want to mention is whenever you see the comparator in vitro, in animals, in humans, we've always used Epidiolex. And I'll come back to why when we talk about the in-human clinical trial. So what we found when we examined the bioaccessibility in cells using CACO2 cells is that we have a very comparable absorption between the Epidiolex arm and the CBtru arm in yellow. And you can see it clearly on this slide. So then, very encouraged by these results, we moved into the animal study. And here I show you the comparator, Epidiolex in blue, and CBtru, the solid formulation in yellow. So the animal study was 4 rats per arm. As you recall, we took all 6 prototypes, 4 rats per arm, 4 rats also in the comparator. And what we looked at was a stable dose of 20 milligrams per kilo of body weight. And what we found here is that CBtru has a comparable bioavailability to Epidiolex in animals. However, the big difference, which you can clearly see on this slide, is the variability between the subjects, which was greatly reduced using CBtru. And therefore, this has been something that told us this is the one formulation we're going to take to clinical trial. And we moved into finalizing the formulation and the powder. So on the right-hand side, you see CBtru. It has a non-crystalline interface. So we started from taking a naturally derived CBD API isolate formulating it into this non-crystalline powder and turning it into what is called the drug product intermediate. So a step after the isolate, a step before the finished medicine. We didn't go all the way to a finished medicine for the simple reason that this has so many different applications. It could be a capsule, it could be a tablet, it could be a powder that you can use in water, many things. All solid formats are possible. Therefore, we developed the drug product intermediate. You can see some of its characteristics here. Obviously, we're looking at an optimal bioavailability with lower variability between subjects, what we expected to see in the clinical trial, but also good physical and chemical stability. A loading of 40%, which was what we reached for our commercial product. That is 4 times higher than what is today commercially available, and dispersible in water, and as I mentioned, can be used in multiple applications. So let's go to the third and last point, which is the in-human pharmacokinetic phase 1 clinical trial. So we took CBtru to trial between September and November 2024 in Canada. It's a phase 1 pharmacokinetic clinical trial. It's a randomized crossover open label trial. So what does this mean? We had 32 registered subjects, equal number of men and women between 19 and 55 years old that took 400 milligrams of CBD, either in a capsule with CBtru or in oil with Epidiolex in a syringe as per the label. And I did mention I'm going to come back to why we chose this comparator. There were 2 main reasons. First one, for a regulatory perspective, Epidiolex is the only FDA and EMA approved CBD medicine, and therefore it is a meaningful comparison for anyone wanting to develop medicines further on and have data to compare to. But secondly, we also know from literature that Epidiolex performs better than most CBD in MCT oil products that are commercially available. And therefore, we wanted to compare with what is the gold standard in terms of bioavailability, because we want to deliver something meaningful for patients. So I'll say a couple of words about the trial design, which you see over there. You see that it was a randomized crossover. So both CBtru and Epidiolex were delivered either with food, a high-calorie meal, or without food. And every single one of the participants went through the 4 arms. So CBtru fed, CBtru fasted, Epidiolex fed, Epidiolex fasted, with a 2-week washout period in between. The primary endpoint was to measure the pharmacokinetic properties, the bioavailability of CBtru compared to Epidiolex in terms of area under the curve of CBD plus absorption in plasma. And secondary endpoints included peak concentration, time to peak concentration, half-time of CBD in plasma, as well as the total drug exposure over time. One last endpoint we included was the metabolites of CBD, because we know that CBD gets cleared very quickly in plasma, so we understood seeing if there was a difference in metabolites, particularly 7-hydroxy-CBD and 7-carboxy-CBD, could indicate additional action in the body, additional effectiveness than what we see prima facie. So this is the design. These are the top-line results I'm very happy to share with you today. So the trial was completed successfully, delivered the first results that you're seeing here. A detailed publication is going to come soon. And it was very promising results because the trial met its primary endpoint, and the CBD plasma concentration time curve over 24 hours after a single administration exhibited with CBtru was higher than Epidiolex in the fasted state and equivalent to Epidiolex in the fed state. Let me say a couple of words about each one of these 2 areas. So in the fasted state, CBtru demonstrated higher, though not statistically significant, bioavailability to Epidiolex, both in terms of area under the curve and peak concentration. When it came to the fed group, Both showed equivalent bioavailability, and obviously the high-fat meal does help with an oil-based solution. So I've included here two graphs from one representative subject. So this is not the full cohort, but it's a very representative subject, and these are graphs we saw being repeated over and over. So you see in green is CBtru, and in yellow is the reference product. You see on the top, on the fasted state, clearly a higher bioavailability, a bigger area under the curve. But there's also one other thing I'd like to point out. You see in both fed and fasted that CBtru peaks earlier than Epidiolex. This is something we saw consistently across subjects and across the fed and fasted states. A couple more observations from the study that I can share with you. It confirmed, as we know from other studies, we confirmed that CBD bioavailability increases with a high-calorie meal. We also saw less inter-subject variability, which means that CBtru may offer a good alternative that is less dependent on food intake and a more reliable uptake of CBD. As I mentioned, the total metabolites were higher with CBtru than with the comparator in the fasted state. And that could actually mean potentially we could achieve the same effectiveness with a lower dose. That's something we're further researching on. And finally, we saw a difference between the sexes, with females showing higher area under the curve than the comparator. But I do have, a higher area under the curve than men. But that could be attributed to the fact that we had the same dose for everyone, 400 milligrams, and it was not adjusted for body weight. A couple of notes on safety and tolerability, very important. Safety was in line with previous studies. Both products were well tolerated. There were no serious adverse events, and any mild adverse events were typical of CBD: fatigue, nausea, abdominal pain, and they were no different between the two arms. We saw no changes in vital signs or weight, and also no concerning shift patterns in liver enzymes or other lab parameters. So, key takeaways. CBtru demonstrated equivalent bioavailability to Epidiolex in the fed state. CBtru demonstrated higher, though not statistically significant, bioavailability than Epidiolex in the fasted state. We confirmed previous findings on the substantial increase of CBD absorption when it comes with a high-calorie meal. Metabolites, the sum of total metabolites measured was higher in the CBtru arm when fasted. CBtru is safe and well tolerated, and it can offer a more reliable and consistent uptake, possibly less dependent on food intake. So we know that we do need to come up with more patient-centric solutions for CBD medicines, and more patient-centric solutions include varied and convenient finished dosage forms, higher API loading, an optimal pharmacokinetic profile, less dependence on food intake, and a more pleasant taste experience. And CBtru can offer that. It is now available for sampling, for clinical trials, and also to be used in commercial products in varied formats, solid oral formats, as I mentioned, some tablets, capsules, chewables, powders, orally dispersible. And one important element that we have been working on is the taste experience, because dsm-firmenich has the ability to combine CBtru with our flavor technology, bitter masking, bitter blocking, and the rest.